So in this video, I'm going to talk about mutation. Mutations are any time the DNA code changes in some kind of way, or altering the sequence of nitrogen bases, and that might have an effect on the protein we ultimately build. So let's talk about the causes of mutations first. There is that natural error rate in copying the DNA during DNA replication, but there are also environmental factors that might add to the mutation rate in cells, and so we call environmental factors that cause DNA damage mutagens. Mutagens generate mutations, and so there may be two subtypes of those. High energy radiation can cause mutations, so X-rays or gamma rays or even UV rays, if you don't protect yourself with sunscreen, might be able to damage the DNA in your skin cells, for example. And there are also chemical mutagens. So a famous example would be the chemicals in cigarettes that can cause cancer when they damage the DNA in your lungs, for example. Okay, so let's talk about the types of mutations then. What kind of broad types of changes might there be? We're just going to talk about two very broad categories in this course. We'll talk about substitution mutations and frameshift mutations, and we'll try to see why those might cause such different effects on the ultimate protein that you build. So let's start with substitutions. I would kind of encourage you to have a translation table handy here if you need to pause the video. It would be a good idea to practice going from DNA to RNA to amino acid sequence. And so I've provided a very short sequence here that I call the original DNA sequence, and then I've highlighted changes that I've made in red in the mutated sequence. So what if this DNA leads to this RNA transcript? And then remember that you split up into your groups of 3 to be able to translate the mRNA codons into amino acids. Let's say you get this. So what happens if you make that change that I highlighted? This would be a substitution mutation because we're just switching one letter for another one while still keeping the same number of total letters, just like substituting one teammate for another in an athletic event of some kind. So if I made that switch, then my RNA code would look like this. And if I split into my groups of 3 again, I would find that CUC, just like CUG before, still codes for leucine. And so I actually have a change in my DNA code, but we call this a silent substitution because the amino acid that made up the protein didn't actually change. And so we're using the metaphor of silence here to indicate that a mutation still happened, the DNA code changed, but it was silent in its effect. And if you look at your translation table, you'll see that usually you can change the third letter of a codon group, maybe without very much effect. For example, CUG, CUU, CUA, and CUC all code for leucine, and so that third letter doesn't matter at all. There are cases where it does matter, like this group here. CAU still codes for histidine, but CAA codes for glutamine. So that isn't always a guarantee, but usually you can change the third letter more than you can change the first or the second. And so let me show you an example where a substitution actually changes the amino acid. What if we change GAC to GTC? Well, that would cause a new codon. And if you look at that in your translation table, you'll see that it codes for glutamine instead of leucine. And so now you have a substitution that does change the protein. How might that affect the protein? Well, it kind of depends on where that amino acid is. Maybe just changing one amino acid doesn't change the active site, for example. Or maybe it kind of has subtle effects. So the answer is it really depends on exactly what the amino acid is doing in the protein. Let's move on to another case where substitution might cause a bigger effect. In some cases, I just changed the third letter of a codon group here, but interestingly, when you look that up in your translation table, you see that it now codes for a stop codon. So that would actually cut the protein off early. And if this were, say, early in the amino acid sequence, and now you've got a stop codon, you won't be adding all of the rest of those amino acids. That will almost certainly cause a protein that doesn't work like it should. And so substitution mutations can cause lots of different effects, from absolutely no effect on protein and therefore no impact on the trait that we see, to maybe a slight change to maybe a big change. Now let's go to frameshift mutations, and I want to argue that frameshift mutations almost always cause big changes on protein. and big changes to potentially to a trait then. And let's see why. If frame shifts involve insertions or deletions of DNA code, not just switching one letter for another. And so you'll see here that when I originally had GAC, CAC, I added an extra C in there, GAC, C, CAC. And so now when I split into my groups of 3, you'll see that I had to shift everything over to the right to create new groups of 3 because of this added letter. And so CAC is no longer together, ATG is no longer together, and they're no longer together in the mRNA codons either. And so what's that going to really do? That's going to change every single amino acid after that insertion because I've shifted the groups of 3. That's why we call it a frame shift mutation. We're shifting the groups of 3, the reading frame, and that's why it causes such a big effect on the protein. You'll see that deletions really have the same effect. What I did in the mutated DNA here is that I got rid of the A that used to be in GAC, and I had to move everything over 1 to the left. And when I create my groups of 3 this time, you'll see that once again, I've got totally new groups of 3, which means totally different amino acids, which means that the protein will almost certainly be built differently and not be able to execute its job. All right. So here's just kind of a quick summary of frame shift mutations. They shift the groups of 3 and therefore will cause all kinds of different amino acids to be put into the protein and will ultimately cause it not to work. So frame shift mutations have humongous effects. All right, so let's just finish this by talking about, it kind of matters where the mutation is occurring as well. So maybe in a multicellular organism, just a mutation in one body cell might not make that big of a difference because even if that cell doesn't work anymore, it might be able to be replaced by healthy cells nearby. The big exception would be cancer. If that messed up cell now continues to divide continuously, then it would cause a big problem because all of its offspring cells would also have that problem. And then the other thing to think about is what if the mutation is in a reproductive cell, like a sperm or an egg cell from mom or dad? If that mutation is passed on to the offspring, then all of the offspring cells will have that mutation because when offspring grow and make up their body of all those cells, it's doing that copying cell division, mitosis. And so the last thing I'd like to talk about is just what kind of effect might a mutation have. I just want to kind of give you the sense for future units that mutations are just the foundation of change in traits. And ultimately, the environment will select what traits are helpful and what traits are harmful. So maybe just a very simple example of a trait of albinism. What if you suddenly have all white fur as a rabbit? That might be really helpful in the snowy forest, but not so helpful in a grassland environment. So ultimately, the environment is going to select what's good and what's bad. Mutations just create change. And so we talked about what a mutation is. We see that there are many causes of mutations and that we ultimately group them into substitutions and frame shifts. Substitutions tend to cause less change to a protein. Frame shifts almost always cause tremendous change to a protein.